right, hi everybody. So this is going to be another new video that I'm adding here for my class. Going to cover, excuse me, sorry. My voice isn't the same anymore after years of teaching. So this video is going to cover cells, their structures, their functions. I'm going to sort of help with how you build a cell using molecules. This is something I normally go over in some of my other classes because I can write on a whiteboard, I can draw it out. So I will maybe briefly cover organic compounds right now, slash organic molecules, and then we're going to think about how we use those to build a eukaryotic cell. Then, as I build my eukaryotic cell, I'm going to go over the organelles that are really important for you to understand what they look like and how they function. Then I'll probably do another video about just talking about the cell membrane and how the cell membrane functions work, especially with how substances move across the membrane. This is an essential concept that my hope is that you come to this class knowing that, but I do know that that is a high expectation. So I want to talk a little bit more about how substances move across the membrane in different ways. And then for my last video, I'll probably do something that I often do again in smaller classes where I talk about how structure and function of cells are related, especially for the human organism. We are made of billions of cells and they are highly diverse. They all specialize, and you want to see how structure and function work over the course, but also you want to start relating that. Excuse me. So we'll start at the beginning. Okay, so let's start here talking about our organic compounds or our organic molecules. Things I talk about in class is, you have this hierarchical scheme, right? This organizational scheme. We go from atoms, which are the basic unit of matter. They have protons, electrons, and neutrons. Their electrons can bond together. So two atoms can bond together because of their electron shell. And when they do, they form larger substances. Those are called molecules. So molecules have to have at least two atoms bonded together. How those atoms bond together depends. You can have two of the same atom, you can have two different atoms that bond together. Now, when you start to make these small molecules that all then bond together into a bigger substance that usually refers to a compound. So, like you can have a molecule of water, and then you can have many molecules of water bonding together to create a compound. For our class, we're going to say molecules and compounds are interchangeable. And yes, I know that that is not true, but it just makes it easier in a bio class to say that. There are inorganic molecules or compounds. And if you remember from class, inorganic means they don't have any carbon. But then you can also have organic. And organic means that they're going to be filled with carbon atoms. These are usually much larger molecules or compounds. They are often comprised of a subunit that gets bonded together repeatedly, often, but not always. So there are four organic compounds that help us build life and help us maintain life. One of those are carbohydrates. Whoa. Sorry, my brain spaced out for a minute there while I was spelling. Then the other one is nucleic acids. You have protein and you have lipids. So carbohydrates, nucleic acids, and proteins, they can contribute to structural components of our cell, but The lipids are going to be the most important group that contributes to how we build a cell. And we'll sort of think about that as we build our cell. So if you want to make sure you know what the subunit is, carbohydrates are made up of simple sugars. Or it's like, let's say bonded together right there. So you don't think carbohydrates equal simple sugars. It is, but it isn't. Okay. Nucleic acids, you have nucleotides. And then I'm going to say, do this double thing here. That means whatever's above it goes below it. So nucleotides bonded together. Proteins. Proteins are amino acids, again, bonded together. Lipids. Lipids do not have a subunit. Lipids do not dissolve in water. That is the reason why they are a group. And this is the reason why we need lipids to help us build our cell. So here's going to be my drawing of my eukaryotic cell. Please forgive my drawing skills with my writing pad. Like I said, I used to use this a lot, and it's been a very long time actually since I used my Wacom writing that drawing pad on my computer, so it's still a little bit shaky. So I got four boxes up at the top here, and I left these four boxes blank, so if you guys follow along, you can use whatever colors you want, but these are sometimes the standard colors that I use because I'm going to color code my cell. So I'm going to make anything in black is going to be lipids. Remember, lipids don't dissolve in water. Then to highlight some of the differences between black and some of the other, between lipids and some of the other organic molecules, let's make one box red. So anything I draw in red, let's make that proteins. It'll give us nice contrast. Then let's do maybe like a light green. You could do a dark green if you want to. It's up to you. Again, you could do whatever colors you want. Let's make that carbohydrates. Then from our last color, oof, like maybe we'll do that in this nice kind of like blue color here. That's a nice contrast. This is going to be nucleic acids. So I'm going to use these four molecules to help me build my eukaryotic cell. What I want you to envision is that this white slide is actually a water-based environment. Imagine primitive Earth, where you basically just had water and a bunch of chemicals. Those chemicals have to come together in some way in order to help us build life. We know this. We just don't necessarily know the mechanism of how that happened or what happened first. But we do know that atoms had to come together. They have to create molecules. Those molecules had to become compounds. And then at some point, those molecules all combine together to create life. In all likelihood, you had to create a boundary first. Now, I'm not saying that's what happened, but it makes a lot of sense. If you have a water-based environment and you need to create a boundary for life, it's one of the characteristics of life, you probably need to have that boundary first so that you can have a sac or maybe something else that's not a sac, but a sac that includes substances inside the sac and substances outside of the sac, thus creating an internal and an external environment. Part of the issue in a water-based environment is how do you do that when you have proteins that dissolve in water, carbohydrates that dissolve in water, and nucleic acids that dissolve in water? You want to use the substance that does not dissolve in water in order to make that membrane-bound sac. So that's the first thing that we're going to do. And I'm going to kind of draw, I don't know. I was trying to think about this, how to do this properly. I've done it a couple different ways over the years, but I'm going to have one kind of box over here and it's just going to stay there for a minute. But I'm going to draw my generic eukaryotic cell, like maybe right about here. And you know what the great thing is? It doesn't have to be perfectly circular. That's perfect for me. So I'm going to take my black and I'm going to draw a circle so that I know that that's the boundary between my inside environment and my outside environment. And then I'm going to, I don't know, maybe I'll do this over here since I have a little bit more room. I'm going to take this and I'm going to put a little box around that and I'm going to blow this up so that we actually draw the cell membrane. And hopefully my Zoom picture is not in the way. Actually, let me do this a little bit differently. Sorry. Zoom does not really show me where I am on the Zoom screen. So I have to kind of think about that. Instead, I'm going to put my box up here and I'm going to blow up my cell membrane right about there so we can draw what our cell membrane will be composed of, because that's what this is. This is my cell, or otherwise known as my plasma membrane. Okay, I like to call it the cell membrane so you don't get confused. Students in the past have asked me, what does plasma mean? It means cell, like it's a cell membrane. So I know I have to use lipids, but if you really think about it, like you have a water-based environment and you throw lipids in there, sure, water and lipids are going to separate because they don't like each other. But how do I make sure you create like this circular or square-based boundary in that water-based environment? How that's going to happen is I'm going to use a specific lipid. It's called a phospholipid. So phospholipids have a polar head or a water-loving head, and I'm going to be real lazy, okay? I'm going to draw my heads as like these squiggle circles. So one layer is going to have my circles that are oriented towards the outside. This is my extracellular part. So that's on the outside. Then the other layer is also going to have heads, but those heads are going to be oriented towards the inside layer. And I'm going to draw it over here just so you guys see too. Then you have your fatty acid tails, which are nonpolar, meaning they don't like water, that are going to orient to each other. So I have this double layer of phospholipids like that. If you're not quite sure what I'm doing here, I'm putting my heads this way, and then I'm putting my heads that way, and my my fatty acid chains go like that. So this becomes what's called intracellular. Okay, so intra means within. So this is telling me this is within my cell, and I have stuff outside of my cell. So this lipid creates a nice boundary so that the inside water stays inside and the outside water stays outside. But then you have to also think about, well, I can't just be lipids potentially. I might need some other things because if you put too many lipids together and the environment gets cold, those lipids solidify. If the environment gets too hot, those lipids are going to melt. So I'm going to put a couple other things in my membrane to help make sure my membrane isn't too solid, it isn't too fluid, and then provide a couple of additional things like, how do I get water? How do I get water back and forth through a membrane that is mostly fat on the inside? Water doesn't like fat, so water just gets kicked out or kicks back in again. So one of the things that I'm going to need is I'm going to need some proteins to help me build some of the structural components of my cell membrane. So what I can do is I'm just going to put like a big red protein that goes through my membrane like that. And you could kind of come over here and draw that like that. You can kind of draw some of your proteins here on your membrane so that you know, yes, the membrane has to have some proteins. Those proteins serve different functions. Structural support, they are for communication, they become cell recognition proteins. Some of those proteins, and this is, ooh, going to be a little bit difficult, so I'll figure out how to do that. Also need to go through the channel. So I'm going to kind of draw what looks like a hot dog bun. Maybe less like a hot dog bun, but it's going to create a channel through my membrane. These channels in the membrane oftentimes are going to allow for substances to move freely back and forth through the membrane. So, like water can be easily transported through the membrane using a protein channel. Salts, like sodium and potassium. I'm just going to come in here with my white and kind of get rid of some of this black so it looks like more like a channel. Um, but that channel is going to allow us to easily move things through that channel back and forth. So sometimes what these protein channels are used for. Then you can also have some other lipids in here. Those lipids are more round shaped. Those are cholesterols. And cholesterols here, what they do is they just kind of push your phospholipids away from each other so the phospholipids don't compact together too much. That's a little bit of what the proteins sometimes do as well. And then lastly was carbohydrates. Carbohydrates is green. You can have carbohydrate chains attached to the outside surface of our cell membrane. So we see some carbohydrate chains attached to the outside surface of our cell membrane. These provide some structure, but mostly what they're used for is cell recognition. So when a cell comes in and recognizes these carbohydrate chains, they're either carbohydrate chains that are recognizing the cell as being part of of your body or being part of not your body. So you see how the cell is starting to be built with these larger organic molecules. Lipids are primarily the thing, but then we also have protein that's going to be used as well. So the cell membrane's function is not just a boundary, it is to allow for um, permeability and really selective permeability through the cell. So some things move through the cell and other things do not move through the cell because of the fact it's made out of lipids. And we'll look at that a little bit later. What I want to do is finish building the rest of my cell and talk about functions of some of the organelles. So eukaryotic cells, maybe you guys know, eukaryotic cells have to have three important things. One of those things is the cell membrane. Another thing is a nucleus. So here you come in and right in the center, you're going to draw in black because we're made out of lipids. We draw a nucleus. So the nucleus actually has two double layers of phospholipids. What that really means is it has four layers of phospholipids to create something called the nuclear envelope, or what goes around the the nucleus. And then it does have a little bit of like protein channels right here inside of that nuclear envelope, again, to allow for substances to move in and out of the nucleus, because you need to move out like RNA, you need to move some stuff in as well. At the very center, in a color that is not on there, I'm going to draw, there is sometimes little circles inside of the center. These are called the nucleoli or the nucleolus. So the nucleolus is the center part of the nucleus where you assemble or you make ribosomes. You basically bring your proteins plus your RNA in, it combines it all together, and then you'll shoot your ribosomes out of the nucleus. So that's our nucleus. Hopefully everybody knows what the nucleus's job is. The nucleus's job is to store nucleic acids. Those nucleic acids are in the form of chromosomes. but you guys probably commonly know those chromosomes as being X-shaped, so I'll draw them X-shaped. Oh, that looks nice. So think about nucleic acids as not being a structural component, it's what's being stored inside of the cell. So that's number two. So we have to have a nucleus. Okay, if you have a nucleus, then what that also means is you have to have an area that is called the cytoplasm that is going to be between the nucleus and the cell membrane. And this is going to be probably a bit difficult because it's not one of, so the cytoplasm is not made out of one of our four organic molecules. That's why I'm going to do this here in like a blue color that kind of reminds you of water a little bit. This is cytoplasm. Cytoplasm is made up of a substance called cytosol. Sol just means solution. Cyto means cell. So this is telling you it's a cell solution. So water plus dissolved substances plus the organelles. That's why it's an area. That's where I'm going to draw all of my organelles. Okay. There are some exceptions to this rule where some eukaryotic cells do not have a nucleus. That includes red blood cells, but they do have a nucleus at some point during their life cycle. So normally all eukaryotic cells have to have a nucleus. Then you can have organelles. In a generic eukaryotic cell, we can draw every organelle and we can say every single eukaryotic cell has these organelles, and that's not entirely the case because in the human body, these organelles are going to be found in some cells and maybe not in other cells. It actually depends on what is the function of the cell. This is what we're going to talk about at the end, how cell structures and functions work together. But for now, we're just going to draw all of those different types of organelles in here, okay? There are two types of organelles, one that are membrane bound and ones that are not. Anything that is membrane bound, it means it is made out of a membrane. This is my membrane. A membrane is always two layers of phospholipids. So the nucleus is actually a membrane bound organelle. Then you have other things. Let's go. Oh, let's do the easy ones first, okay? Because you guys all know some of these easier ones. So I'm going to draw a little like bean with a little squiggly on the inside. Those are mitochondria. So mitochondria are actually two layers of phospholipids, one on the outside and one on the inside. And everybody knows what they do. They make energy. They're the powerhouse. They help us provide the cell with energy in the form of ATP. So it's one organelle. Then you also have a set of organelles that are called the endoplasmic reticulum. The endoplasmic reticulum is a membrane-bound organelle that essentially comes off of the nucleus right here. So it's like an extension of the nuclear envelope. And they are a network of tubes. So they're hollow tubes that are attached together. There are two endoplasmic reticulums, okay? One endoplasmic reticulum is a rough endoplasmic reticulum, and the other one is a smooth endoplasmic reticulum. And you'll have to forgive me, I've never been able to draw a smooth endoplasmic reticulum like the artists do. It just doesn't make any sense to me. But they are connected together because they're both made out of a double layer of phospholipids because they're endoplasmic. So endo means inside, plasmic means inside the cytoplasm. The reticulum just means it's a network. The rough endoplasmic reticulum, you'll know it because it's actually going to be studded with ribosomes. And ribosomes are non-membrane bound organelles that are primarily made of protein. So I'm going to draw a couple of ribosomes right here. They do have some nucleic acid. It's like nucleic acid plus protein, and that's hard to sort of draw and understand. So I'm going to draw it as like blue with, oh, I did the wrong blue right there, did I? Yeah. I was going to do a red plus blue together. Maybe that just is too much, but you can draw some what are called cytoplasmic or free ribosomes right here. And if you want to think about it, also has some nucleic acids, and that's why I'm going to do it in both red and blue. But it's a lot easier to tell everybody that, well, that's just made out of protein. Okay, so these are my ribosomes. So I'm going to do the same thing on my rough endoplasmic reticulum. I'm going to draw on top of the membrane some of these little red dots, and those are going to be my ribosomes. And I'm going to forego the blue so I don't spend a whole lot of time. Okay, so that's my rough endoplasmic reticulum. The rough endoplasmic reticulum and ribosomes, they both make proteins because their job is to make proteins. When you put that on top of that network inside of the cell, it also makes proteins that get shipped into little packages, or I should say boxed into little packages. These little packages kind of bud off right about there. These are called a vesicle. And then the vesicle takes those proteins and they ship them to the next place where they need to be modified. And maybe I should have put my thing up over here, but that's okay. The next place that they ship them to is a part of the cell. It is called the Golgi apparatus or Golgi. That's what that is. So again, Golgi is it's a set of membrane, so double layer phospholipids that kind of have this curvature that curves towards the cell membrane. And what Golgi does is it modifies those proteins that it gets from the vesicles. So these proteins go over here and modifies them, and then it reships them so that the proteins can be released outside of the cell. These proteins could be whatever the cell needs. It could be like collagen, it could be keratin, it could be insulin, depends on what the cell is. So you see, that's also made out of lipids, okay? Protein is on the inside of it. It's not making it. It's just that proteins are being made inside of the membrane. It doesn't make up the Golgi membrane. Then over here is my smooth endoplasmic reticulum. I'm just going to abbreviate ER, even though I've gotten in trouble for that before, because my writing pad is not working so well today. So the smooth endoplasmic reticulum, what it does is it makes and breaks lipids. So if I need more lipids for my membrane, if I need more lipids for my nucleus, if I need more lipids somewhere, then it will help me make those, but it will also help me break them apart. So some cells that might do removal of toxins, especially lipid-based toxins, will have more smooth endoplasmic reticulum to help them with that breakdown of those lipid-based toxins. So you got mitochondria, you got rough endoplasmic reticulum, Golgi, vesicles. Sometimes you have things like peroxisomes, you have lysosomes. Peroxisomes, lysosomes are all types of vesicles that just store different things. That's really what they are. You can have a vacuole. A vacuole is just a larger vesicle that stores things as well. Oh, let me see. Did I get everything? I feel like I'm missing something. There is one additional molecule, or sorry, one additional organelle that is not made out of lipids. It's actually made out of proteins. These are called the cytoskeleton, and sometimes you can see them in the cell as just these long dashes. Oh, sorry. It's just, I need to update maybe. So cytoskeleton are these long filaments of protein, and they provide things like structural support for the cell. Here, it's good that I put that protein right there because the vesicle walks along the cytoskeleton. If you have anything on the outside of the cell, like you have cilia, those cilia are actually made out of cytoskeleton. You can have a long flagella made out of cytoskeleton, and that produces movement for the cell. So you want to know what are these basic organelles, you want to know what the function of the organelle is, and then you want to think about, well, if I have a cell that does this function, what organelles would my cell need in order to do that function? This is how structure and function are interrelated and how you can use structure and function to understand things better in biology, even especially in an anatomy course. So I'm going to think about this because maybe there's one or two I missed here, and I might come back to it later. But then I want to go over this whole idea of like the membrane transport and look at the membrane one more time and think about how we can move stuff through the membrane in different ways because it will affect the health of the cell if you put too much stuff in or if you take too much stuff out. So we have to have that selective permeability in order to maintain the health of the cell.